whenever you want, please.
Okay, let me check. Is this working? It is, right? Okay, good. Oh yeah, sorry, Robocop. Okay, creepy stuff. Okay, good morning everyone, and thank you for being here the last day. As you've noticed, I've been telling you the story of researching criticality from my lab, essentially, and in chronological order. So today I'm most excited because I'm going to show the latest results, the results that I like the best, actually, not only the latest. And great, so, but I wanted so I'll start today connecting with yesterday. So yesterday, we were trying to understand what was going on in this data with urethane anesthetized rats. I hope by now you agree with me that telling you which anesthesia it is important. And just wanted to thank you. I'm going to talk about mostly Danielle, Gustavo, and Caio's work today. But data was collected by a bunch of people, like I mentioned yesterday. So I was showing you results, this kind of data, which was what you get for urethane anesthetized rats. So to recap, we have this huge difference in behavior spontaneously. So the rat can go to this very asynchronous to this more synchronous behavior. And what we did yesterday was parse this data by this level of synchronicity of coefficient of variation, and then run avalanche analysis. And depending on the level on your CV, on how synchronous or asynchronous you are, this exponent changes, and this exponent, this third one, also changes. And then you go to statistical physics, to theory, to inform you that they're not independent. They should be, they should satisfy some scaling relation, the so-called Crackling noise scaling relation. And since this left-hand side, the right-hand side depend on CV, you try to see whether they match at some point, and they do. And you get happy because you take this as a signature of criticality. And right? And this also works for other data sets. And I try to look at exponents that are populating this plot here with the tau t and tau, and you put the tau t and tau on a scatter plot. They scatter along a straight line, curiously. And if you look at the place where the scalar relation is satisfied, you see that the exponents are not exactly mean field, they're hypercolation, therefore suggesting that a different model was required. So this was part of what I told you yesterday. But then, of course, Tawan Carvalho, doing his PhD, showed us that we could be completely wrong, perhaps, I don't know. Just taking a full, perfectly mean field hypercolation model with classical exponents, then subsampling it, and once you subsample and subject the simulation data to the same pipeline of analysis, you get this wide distribution of CV, even though you're changing a control parameter very little, and you have all these funny exponents which are not mean field direct percolation, even though the model is direct percolation. It's just distortions out of the procedure. So your analysis is kind of morphing things, and this explains data very well, except for this behavior, long-term behavior of the coefficient variation of the data. Right? So when you look at the experimental data where this crackling noise scaling relation is satisfied, and therefore where you should expect, you would predict that the system is critical because all the things are satisfied, and then these are the exponents in the spread of exponents. Data and this subsample model agree, but you know that those exponents are not real in the sense that that's not how the model is working. You know that exponent tau is 1.5, and you know that exponent tau t is 2, so this is the black dot here. So this distorted result However distorted the exponents are, it still predicts that the system is critical, but only if the model really is near criticality. If you are 3% off criticality, you don't get these results. And so I was telling you yesterday that you have the real coupling parameter of the model really close to the critical point, and still you get this very wide distribution of CV, therefore suggesting that CV, that in the previous paper we interpreted as a control parameter that we not control, so turns out to be a very lousy control parameter, not a control parameter at all. So while the experimental results, the way we interpreted them, seem to suggest that you are criticality en passant, if you believe the model, then the model is suggesting that this whole range of CV, along most of which you have these signs of criticality because the model is near the critical value, suggests that that's not the only way to interpret the results, but rather that you could be all the time near criticality. So this is where I stopped yesterday, more or less. No, actually, well, and then if this is the case, then these very synchronous and asynchronous states will be fluctuations around the critical point, which is kind of a bold proposal. And since here we compare experimental results with model results, well, we would like to test these ideas experimentally without resorting to a model. Can I go back to this data and try another way of analyzing the results to see whether this idea holds? One thing that I showed you yesterday was the maximal entropy approach, in which if you take the whole data, you do get these nice signal of criticality. By the way, I forgot to add the reference here, if someone wants to have a look. And if you parse by CV, you get, again, a result which suggests that, yes, near some specific intermediate CV value, you do have a signature. Okay. So it's puzzling for us. I imagine that you're puzzled as well. So I'll like to come back here. Can I probe this experimentally in a different way? So this is what, both approaches were based on average analysis. I would like to do away with this thing for a while, perhaps for good, and try a different approach, which was in Daniel Castro's PhD work. He's now a postdoc in our group and is moving to Germany next week or so. So we'll do the normalization group approach that Bill Bialek proposed. So to remind you, if this is an Ising model with coupling between neighbors, and if you, with some coupling J0, J0, and if you take the average of these guys, you can mathematically, in a model, rewrite the coupling J1s between what these super block spins will be. And so when you do this mathematically, effectively what you're doing, I hope you can appreciate in the picture, is that you're changing the scale at which you're watching your system, right? If you look here, you lost microscopic details that were here and are no longer here. You cannot see them anymore. I mean, mathematically speaking, I'm saying. And now you take this system and do this again, and you have this J2 with super spins and stuff like this, and you go on, right? So as you mathematically proceed with these ideas, eventually you reach some kind of fixed point. And if you reach a fixed point by changing scale and nothing changes any longer, that almost by definition means that you reach some scale invariance, because you're changing scale and things are not changing anymore, mathematically speaking, right? This is the more fancy plot, which I took from Michael Fisher's 98 review, in which you have this, again, this temperature and external field. You have a critical point here somewhere, and as you do this transformation, this thing gets morphed, but eventually there's a fixed point here, separating two different trajectories, like one disorder phase, another phase in the simplest case, and this fixed point in parameter space here it's a very special point. You expect scaling varies and non-Gaussian distributions like Bill told us the day before yesterday, if I'm not mistaken. Okay? And of course, this is, like Bill mentioned, this is the gold standard in theoretical physics for understanding phase transitions, the renormalization group. In models, that's the point. The thing is, that should be apparent by now, we don't have a good model of the brain. That thing, by far, we're very far away from it. So, yes, this is it's a beautiful accomplishment, intellectual accomplishment for humankind, which was give the Nobel Prize to Kenneth Wilson. But if you don't have a model for the brain, well, enters our dear friend Bill Bialek with two very nice papers with Serena Brad in Journal of Statistical Physics called it's PCA meets RG or the other way around. I don't know. Well, RG meets PCA. Anyway, PCA. Anyway. And then there's the other one with Lino and Mechelin, PRL, and collaborators. So this phenomenological renormalization group, as people came to call this technique, is data-driven and model-free. So now we can talk, because now we have lots of data in neuroscience. So the idea that he already told you, but I'm going to repeat because I want to use this technique throughout my lecture today. You take, like, this is from their paper, from their 2019 paper, different units, let's say neurons, and this is fluorescent signals. You bring together those who are most correlated, 1 plus 2, and then 3 and 4. They are ordered according to how correlated they are here. And then you do this again and again and again. You couple by correlation. Since you don't have the interactions in the Ising model, which can inform you who's connected to whom, you replace it by a proxy, which is correlation, and see what happens. And as you do this, you observe whether observable scale non-trivially with cluster size, and whether normalized activity converges to non-Gaussian distributions. Okay? So this is what we refer as to real space phenomenological renormalization group. And as an example, Bill showed you already this previously. You have the probability of science, or a minus log of probability of science should scale with cluster size as a power law. So this k to the power beta, this beta exponent governs this thing here. If you imagine that you had a Poisson process, right, with some rate, and another Poisson process, say another neuron, and they're completely independent. One property that we know about Poisson process, you join the two, it's still a Poisson process. When you sum the rates, right? So if you have n neurons, you have with more or less the same Poisson rates. You bring them together, it's like one Poisson with whose rate is like n times larger. Therefore, it will be proportional to n. Therefore, beta equals 1. In that simple case, so beta equals 1 is like the control. Nothing interesting is happening here, kind of value. Okay, and here they're showing this normalized activity converged to something which is not Gaussian. The easiest to understand is like Bill explained to us, like the variance. So if those things you are summing are not correlated with each other, then you have the central limit theory telling you the variance goes with 1, exponent 1. System size, the power 1. If they're perfectly correlated, it goes with power 2. And in data, they go with some intermediate exponent. Correlation time, as increase your system size, also increases with some power of the system size, which, as Bill mentioned, yes, is a very small exponent, z. And you can look at the spectrum of eigenvalues and fit a line here in the beginning, and this exponent mu is another exponent. So 1, 2, 3, 4 exponents, and then this non-Gaussian distributions. I'll focus mostly on the distributions, actually, and particularly not in the real space renormalization group, but in the momentum space phenomenological normalization group, which works like this. Suppose you take this raster plot of many neurons. We take more than 128, and you take the, so I'm not going through the details of why this is so, but this is in this PCA meets RG, RG meets PCA Bill paper 2017. You take the eigenvector of the covariance matrix, you build projectors onto these subspaces, and you sum the projectors, but not all of them, just a couple of them. So if you were to project only on the largest eigenvalue, this is PCA 1, so the first component of PCA. If you project on the second component, etc. But here you're summing, and take, for example, this, what happens if you do this to this blue neuron? So if you project into all the eigenvectors, you have the same thing. The resolution is kind of bad, isn't it? And you take half the eigenvectors. Strange. Isn't it strange? The resolution is odd. But anyway, you'll see that it shouldn't matter because both the screen and the procedure is cross-graining the data. Okay? And n over 8, n over 16, and you see that you're losing, you're throwing away, not losing, you're just throwing away supposedly irrelevant details by construction, right? Because you're just keeping the largest eigenvalues. So there's a n over 16 largest eigenvalues. N over 4, n over 8, n over 16, n over 32, whatever. So this is activity, renormalized activity, of course, grain activity of this blue neuron. Now you do this for all the neurons, and you collect the probability distribution of this thing, right? And what you get in some cases, like the data I'm going to show you, is this blue distribution here. You can see here, if you can see anything at all, actually, from n to the n over 4, n over 8, over 16, that is converging to this blue distribution here, which is non-Gaussian, right? If you shuffle these spikes here in each neuron, keeping the interspike intervals, and redo the procedure, then you get something which is way closer to a Gaussian. So this is revealing some structure there and points to that idea of a critical point. One way to characterize how Gaussian or non-Gaussian this thing is, just by looking at kurtosis of the distribution. This is the kurtosis. Here you have 12 essentially for the real data and 6 for the near Gaussian. You could protest it, but isn't it true that for Gaussian kurtosis is 3? Yes, for a perfect Gaussian. You don't have a perfect Gaussian here. You have this thing here, this difference here, and apparently this is enough to shift from 3 to almost 6. Okay, just it took us a while to convince ourselves that this was the case, but apparently it is. Right, Gustavo? Gustavo is here. Yeah. So we want to check, to control whether this thing is how non-Gaussian it is by comparing with what you obtain from the same data just by shuffling. Okay, this is our control. So here we have a kurtosis which is for this 30-second window, which is, I think, larger than this surrogate kurtosis, and we'll come up in a minute with some criteria. There are, however, in other windows for the retained data, for which this is what happens. Here, is this red curve sufficiently non-Gaussian to you or not? It's debatable, right? Yeah, this is non-Gaussian, but well, the green one is non-Gaussian either. It's not. So there's a thing there about how to compare stuff, okay? So suppose we revisit this problem here of the retained data. First, forget about CV for a while. Let's have a look at the whole data, okay? The whole data, just and run this PRG stuff. You do this thing, you connect things, and you do you do the coarse graining in real space and in momentum space, and you have the mean variance. So Again, I don't know what you can see. It's kind of odd. Here is the independent value. No, sorry. So this is the real value, and this is the shuffle data. You get alpha equals 1, and for the real data, you get alpha 1.53 or so. Beta also has 0.63, so non-trivial behavior. Z is a kind of noisy exponent, but again, for shuffle data, get Z essentially 0, and for the real data, 0.39. You get a nice scaling here also for the eigenvalue, so mu equals, I cannot read, there. I cannot read here either, but 0.56, I think. And most important for us, the probability distribution of the coarse-grained activity is clearly non-Gaussian, right? It has a asymmetric peak here, and it converges to this thing. Here we have a lot of data. We can go up to n over 64 modes. And whereas if you shuffle the spikes before you go here, you get things which look pretty much Gaussian. So this is nice, for starters. But we arrive here informed by the neuroscientists that CV was an important thing because it tells you about synchronous and asynchronous behavior. So we go back and reanalyze that thing, parsing the data by CV. Oh, sorry, yes, I'm some blah, 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 the names of the exponents. So we return to the data, parse the data by CV, and now redo this by CV and see what happens, okay? So again, I have another plot of the same thing. So this is CV as a function of time, orders of 2 or 3 hours experiment, depending. And here you have a distribution of CV for this animal. You'll see that each animal has a completely different CV distribution. Here again, examples of low CV, so highly asynchronous behavior, raster plots, more synchronous and intermediate CV value, and a high CV value here. And you run the phenomenological renormalization group for these different CV stripes. Oh boy. Can you see anything? You can, right? So color-coded here is the CV value from darker ones being lower CV to brighter ones being higher CV. And you see for the darker ones, you have something which looks more or less Gaussian. And as you increase CV, the shape of distribution already at the maximum coarse graining goes to something highly non-Gaussian. And this is true for all animals. So what we would like to see is a plot of CV in the horizontal axis to something in the vertical axis which tell me how non-Gaussian the thing is, and we're choosing kurtosis again, right? So the plot I'm going to make is CV as a kurtosis in the vertical axis of this renormalized distribution as a function of CV, and you have this very nice behavior like this. The gray stripes here is the kurtosis values that you expect for that you obtain for surrogate data, shuffling all the spikes, and therefore we take actually the upper part of the gray stripe here is like the mean kurtosis of surrogate data plus 1 standard deviation. So z-score equals 1 or something. If you do z-score equals 2, it doesn't change much because the standard deviation is kind of small. So, right? So what we use in this paper is judging from this plot, is a criterion for scaling behavior. We say that system has non-trivial scaling if the kurtosis that you obtain in the normalized distribution is larger than this surrogate value plus some standard deviation. Okay? And you see that there's no non-trivial scaling if you're below. It took me a while to retrain my brain when I looked at this picture, because when you look at this picture, as a physical physicist, we are, I don't know about you, but we're talking about phase transitions, criticality. We think about control parameter as a function of order parameter. Let's go like this, right? Or something like this. And this look like, but this is not that, okay? That's not it. Because what I'm saying here is not, please, this is important. I'm not saying that this is a critical point. No, this whole thing is supposed to be critical, because this whole thing has non-trivial scaling. So, sorry. This whole CV region amounts to critical behavior. Therefore, connecting with yesterday's results for the model, which predicted that you have a small region parameter space of the real model, which gives you this wide variation of CV. So CV is not a control parameter. So everything here is giving us a signature of critical behavior for a whole range of CV values. Daniel Miranda made a nice movie in which this plot here, you can see this is long time, I'm sorry I missed the units here, but this is like 2 hours of experiment. CV is changing, and as the CV goes up, this renormalized distribution of activity becomes highly non-Gaussian. When CV goes down, it becomes more Gaussian. And this is true for every animal, except that each different animal will have a completely different history here. They all satisfy the same thing. Okay? Is that clear so far, what I'm trying to tell you? Yeah? So this is what I was talking about. So this scenario here of this whole CV range yielding with the phenomenological renormalization group technique, a signature of critical behavior in a broad region of CV values agrees with what the model predicted yesterday, right? So perhaps all the time is a little too much, but most of the time near criticality. In fact, you can go back to look at exponents, except now we can be more careful. Not look the exponents alpha, beta, z, and mu, not in any case, but just looking in this region here where you have an additional signature of criticality, and being careful when the system is here, because you know that here you're converting to a Gaussian. So whatever you fit in your exponents, maybe don't need much. So I'll take this axis of kurtosis, put it in the horizontal here, so you see these gray stripes in the vertical line, like this. So this kurtosis horizontal axis. 2 small kurtoses amount to non-Gaussian behavior or close to surrogate values. And you see that as you fit alpha, things here get all over the place. But as soon as you pass this threshold, when things becomes significantly more different from a Gaussian in the distribution, then the exponents have, well, more or less stable values. Well, it fluctuates a lot, but still, right? It has some value here, which is non-trivial, 1.7 for alpha. Beta, the trivial value is 1, and you're away from it. Mu has no trivial value that I know of, and it's more or less stable here. And Z, always with small values, but still significantly larger than the trivial value, which is 0. And you cannot see, I'm sorry, I don't know what's going on with this projector today, but in the inset, you have the goodness of fit, which become very good as once you are in this scale regime for sufficiently large kurtosis. Okay? So once you take care of throwing away these exponents which are varying a lot, and take only those guys, you can take the average, standard deviation, whatnot, And so different bars here are different animals. Exponents are more or less stable across these animals. You can take averages, population averages, and standard deviations. The gray stuff here is for surrogate data. So they're very different from surrogate data. And you can do something else. You can attempt using this it's an arbitrary criterion, but I think it's good enough, that saying, okay, if the kurtosis of a given 30-second window is above surrogate values, then there's scaling. Otherwise, it doesn't. I mean, I'm just deciding like this. So I can revisit all my windows for all the rats and just count. Okay, so how many of them are scaling? Which portion? How much time does the system spend in this critical behavior according to this signature? So I'll come to that. So first of all, here I'm showing you this kurtosis as a function of CV curves, like the one I showed before in this upper right hand of the plot, for all the different animals. So they are essentially the same. They all depart from non-trivial behavior here, CV 0.7 or so, and increase. And the way they increase here is different for all the different animals. And this here is surrogate data, okay? And it doesn't matter. To me, the fact that if they are above some surrogate values, that's enough for me to call it non-trivial scaling. This curve here, kurtosis as a function of CV, across all animals, they have the same qualitative shape, despite the fact that the distribution of CV for different animals is completely different. So like I mentioned before, you anesthetize a rat with urethane, the way its coefficient of variation, population rate, how much synchronous or asynchronous it will be during a 2 or 3 hour experiment is completely unpredictable. You don't know what's going to happen. It can be anything. Some rats, like this orange one, tend to be less synchronous a little bit. These green ones tend to be more synchronous. The only thing is know that if it's too much asynchronous all the time, your anesthesia failed. You should do something about it. That's all the experimentalists tell me, okay? But other than that, despite this mess, this completely different histories along the experiments, once you organize them by the kurtosis of the coarse grain activity, they have this behavior here. So CV here has been a useful quantity in a way. What it's telling me is that low CV values, that is, more asynchronous behavior in the urethane data, are typically do not show signals of criticality. That's very interesting, because in a couple of minutes, I'll come to freely behaving, not freely behaving, but non-anesthetized animals doing something. And as you perhaps remember from my cortical states review that I showed, if the animal is doing something actively, its activity is asynchronous, right? So electrophysiologically, if you look, it looks like this. I'll come to that. Once you have the probability of CV and you know how to calculate whether it's killing or not, given its kurtosis, you can do things like this. So the probability, once you decided that a window has scaling, what is the probability of CV that it has a given CV and has scaling? Well, this is the probability of CV, and if it has no scaling, this is what you get. So the no scaling windows tend to have this lower CV, and the scaling windows tend to have this higher CV, with some overlap between the two here. This is what this hash region means. And then you can calculate the probability of scaling given the half CV, given the value of CV, and it's an increasing function like this, which saturates at 1 as a half height measure about CV 0.7. So what this technique is telling us that for the urethane anesthetized animals, first, if you have a large enough coefficient variation, I mean, synchronous enough, which is not very synchronous actually, 0.7, activity, you're reaching scaling signals in the coarse grain activity. And if you count all of them, I can put a number to how often you are critical or you're visiting a critical state, which is 76% of the time. Okay? So this was nice. Of course, you're entitled to be skeptical if you want, I don't know. I mean, if scaling, as observed in this technique, amounts to criticality, I don't know, then the cortex under urethane goes in and out of criticality, because then sometimes it goes sufficiently asynchronous and uncritical, and then it comes back. But 76% of the time, it is in a scaling regime. So these results, as how I interpret them, on the data, just support the prediction of the model that I showed you yesterday. So this is nice. So is a way of checking that assumption that, or the interpretation that came out of the model without a model, right? So a wide range of synchronous or high CV states are manifestation of single critical point. This is important. So the critical point is here in this kind of plot. And in the other kind of plot that I showed, which is kurtosis of the renormalized, the coarse grain activity as a function of coefficient variation of the data, which is more or less like this. This is critical behavior, not this one. It's not here, it's here, okay? The exponents I got from here are not obviously related to classical exponents in statistical physics. Therefore, the caveat is that I have no information about the universality class. And by now, after having tried, have probed this mean field direct calculation universality class, which is something that people still use to this day to think about this problem, I'm not sure I care too much about it anymore. Maybe I will in the future, but if I have a well, you follow the path of my lectures here that people started with avalanches, and then I'm showing that avalanches are problematic in different ways all the time, right? Gives you false results, you have to go back, reinterpret them. It's messy. I find this way cleaner and less prone to the issues that we face with avalanches so far, I don't know. And again, I call your attention to the fact that, well, I'll show this in a minute, but PRG on non-anesthetized mice, which I'm going to show in a minute, does show signals of criticality in asynchronous states. So not all synchronous states are born the same. And how to differentiate them? But apparently, this technique already does that for free, right? But it's interesting because if you look at the data, say, awake animal doing something, I show the raster plot only, I don't show the animal. It's asynchronous behavior. I parse the data from an anesthetized animal with urethane in the low CV regime, it's asynchronous as well. Show those two plots to an electrophysiologist and tell them, well, which one is which? It'd be hard to tell with other techniques. This technique separates them completely, as I show you now, I think. But before I do this question here, as you can imagine, was rounding in our heads because why? Because we had these previous results with nice avalanche scaling results, experimental data, and then you run a model which kind of destroyed the initial idea, right? So here we just wanted to check. So Caio, one of our master students, They said, well, let's run models that we know the critical points of, and run the PRG assuming we didn't know. And then the excitable cellular automaton model that I showed in lecture 2, if you run as a function of sigma here, have a critical point at 1, and if you blindly run the phenomenological renovation group analysis, its kurtosis will peak at the critical point. And if you run the other model that I showed with excitation and inhibition, the probabilistic-like Brunel model, it will also peak even better here at its predicted critical point. Interestingly, the kurtosis peaks more sharply than, say, alpha exponent, which has some broader distribution. So if you're not exactly at the critical point, but around it, you still get some non-trivial kurtosis, and you get some non-trivial values of exponent here. So the exponents, I'm just choosing alpha here, non-trivial alpha here, according to this model, can mean that you are not exactly the critical point, but maybe near 1, for instance, right? Which is probably good enough if you are a criticality person who wants to see, well, do I have indications that you are near a phase transition? Near is, to me, is good enough. For some people, it isn't, I don't know. Okay? Well, so apparently, as far as these models go, the answer is yes. Now, let me go back to my lecture 2, when I first invoked these ideas of brain criticality. Why would you expect the brain as a dynamical state to operate near a critical point? Well, because supposedly it will give you a lot of interesting properties that would amount, presumably, to functional cognitive advantages. That was the whole point. The thing started in 2003. And the problem is that I've shown you essentially results for slices, anesthetized animals, and when I showed you freely behaving animals, they were not doing quite anything. The signatures of criticality were kind of messy, and we couldn't ask them if they were feeling any functional advantages being critical. So critics of criticality correctly point out that this connection between this highly advertised connection between brain criticality and cognitive and functional advantages were never there to be found in the first place. Where are these advantages? Experimentally speaking, of course, look, I can predict that dynamic range is maximized in a model, nice. And then you go to a slice and see that this is more or less true. Yeah, but this is still a slice, right? Which is doing nothing. And you go to somatosensory cortex of an entire animal. Yeah, but it is anesthetized. Yeah. So look, I like these results. I appreciate them very much. And I was happy for many years when they come out because someone bothered to do an experiment just testing our theory. But still, so can brain criticality be linked to an enhanced performance in a task experimentally measured? And this is what Gustavo, who's here, is tackling in his PhD, okay? So we believe the answer is yes, and that's why I'm so excited about these results, because I think it's, after 20 years in this literature, I think it's the first time that this has been demonstrated, okay? Before we jump right into the results, I think it's just, I was inspired by the coffee machine earlier this morning that people showed me has several different impressive features that we didn't know of yesterday. So it's, you can show, it's like a NASA coffee machine that we didn't know. Let me show you how electrophysiology technology changed in 20 years. So the first experimental result that I showed you, apart from Hubble and Wiesel and stuff like this, right? And from the turn of the century, you have 6 electrodes in vitro like this. Nice. State of the art today is these, apart from the optical recordings, okay, that Bill mentioned with the photomicroscopy. But as far as electrophysiology goes, you have these neuropixels probes, high-density recordings. You have here 960 recording sites, out of which you can choose 384 configurable channels in the software once you implant the electrodes. So here are the scales, right? I remind you that a body cell in the neuron is like 10, 15 microns. So these were in vitro recordings. These things you can, of course, do in vivo, and you have a lot of channels here, and you can put several of these needles onto the mouse head. So here's a picture, not from our lab yet. This is from the Allen Institute, data from the Allen Institute, which is amazing data. If you work in neuroscience, you know what I'm talking about. If you don't, I'll mention briefly. So this is one. This is a glass or plastic cup on the mouse on the head of the mouse. This is 1 millimeter hole. Here you see this black needles, which are these guys here, which are already implanted in the mouse brain. And this is a picture from above. This picture with the probes here. You can barely see the probes in any device, but they're here. And this is a cartoon of the experiment that the Allen Brain Institute made. So you have the mouse are awake, they have their head fixed, and they have this running wheel so they can run however they want. And here you can place some visual stimuli in the monitor, and on top of that you can monitor loss of behavioral data. For instance, you can put a camera to their eyes so you can track how their pupil are dilating or contracting, because this is a sign of how attentive they are. It's a well-known behavior signature of attention and engagement. And you can monitor wheel speed, blah, 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 right? And then Bill showed you this AI software, which you can mark the parts of the animal and then track their behavior. You can film them while they're doing this and look at the palms, look at parts of the body. Okay, the head is fixed, but still. So a whole range of behavioral data can be obtained with this kind of setup. This is a cartoon of the experiment. The real experimental setup is here. So, still this is a paper mouse, but this whole apparatus is here. So, the Allen Brain Observatory released this data in 2019, I think, the NeuroPixels visual coding. That's the experiment from which we're taking data and analyzing the results. Before I do this, please note the name of the place. It's a brain observatory. So the landscape is changing a lot. So these guys have a lot of money. They do impressive experiments, sophisticated experiments, large experiments. The things I'm going to show you, they did for how many animals again, Gustavo? Perhaps 100 or 102 mice. Very sophisticated experiments for 102 mice. Very carefully documented and stuff like this. And once they have it done, they make it publicly available for everyone to grab, right? So this is almost like astronomy a little bit. That's why it's called an observatory. And I'm under Brazilian conditions. Little money, lots of red tape. I'm trying to buy one of these NeuroPixels, and because I want to do some experiments like this. But we keep having this discussion in our group. Like, should we? I mean, why? Because those guys are, I mean, we could not compete with this, right? We never do something like this. But we can do other stuff and ask different questions. But if you are into neuroscience, you want to analyze data, and you don't have a lab, perhaps you don't need one. So the panorama is changing in neuroscience, and it's changing in a good direction, I think. It's very nice. So if you're interested in neuroscience and want to join our efforts, please come. There's lots to be done. So this kind of setup allows you to do, please try to remember Hubbell and Wiesel experiment with a cat back in the 70s, right? You listen, and it was one electrode. This is what you get with the neuropixels. This is not data from the Allen Observatory. It's data from the University of Arkansas, Woody Schuh's lab, where Antonio Fontanelli is working now, and he kindly sent me, as a courtesy, the movie from their lab. So you have this one neuropixel probe in the motor cortex, and you're monitoring this mouse. In blue, you'll see the wheel speed, and in red, the population activity of all the some neurons, but you'll see the activity of all the neurons in real time. So this is kind of recording that you can have, I mean, many labs have nowadays. Our lab is going to try. It's a pity that you cannot see the colors here, but each line here is a neuron, okay? So the animal stops, activity, population activity in the motor wheel, so it starts moving, then you have higher spiking rates, et cetera, et cetera, et cetera. And they have these movies, they can then feed the AI software to monitor palm speed or whatever it's doing with its whiskers, et cetera, et cetera, et cetera. So I think it's nice to illustrate how fast things are changing in this field. I mean, from a point of view of recording technology and what you have available and the amount of data. So there's lots of data. It's very nice. Okay? So back to Gustavo's work. So this Allen Brain Observatory Visual Behavior Neuropixels experiment, you have this highly trained mice. So before they go to surgery, before they have this thing implanted, they are well trained in a task. So they get used to having their head fixed, even without the electrodes, and they're trained in this task. What is the task? It's a visual task. So you have this images, realistic images, which are flashing on the screen every, I don't know, 250 milliseconds, and then has some spacing, another one, etc. And they flash this image, and then they flash it again, the same image, and then they flash it again, the same image, at a much higher rate, of course, and then they flash it again, but a different image. So when they change images, the mouse can be rewarded if he noticed the change, and that he can lick. There's a lick port here, in which he gets, I think, water or some juice or something that he likes. Okay? If he licks in the wrong time, he doesn't get anything. And if he starts licking this all the time in order to be smart, the software won't change the image. So don't do this. So, they have him pre-trained, and they're very good. As I'll show you in the later slide, they get this right like 70% of the time, roughly speaking, on average. Okay? Pretty smart mice. So this is the test, detect change of images in a sequence. And they lick respond and get rewarded through this lick port. So, like Gustavo mentioned, reminded me, they have over 100 animals, but we chose to analyze signal criticality in V1, primary visual cortex, because it's a visual task. So if criticality serves any purpose, that's where you should look, we thought, right? And we selected those recordings which had more than 128, which is 2 to 5, 6, 7, I think, to the 7 neurons in V1. So that left us with 62 sessions of 44 mice. Yeah, because there's that too. They repeat sessions for the same mice. So we can compare, for instance. I don't know. Okay? How do we assess criticality in these data sets? So I'll show you, well, with phenomenological normalization group, like we did for urethane data. So this is V1 in the rats. You have the neuropixels needles going in and recording many other regions, but I'm going to only talk about V1, okay? So they have the 3 phases of experiment. The active phases, which I've just described, in which we have natural images. The animal can move their paws if it wants, and gets rewarded when the images change. For 60 minutes, they do this, 1 hour. And then the animals perhaps get some rest, I don't know, in which it is presented with these Gabor patterns, which are stripes on the screen. Why? Because this was Hubel and Wiesel did years before, so people knew that from before the V1 codes for these things. In anesthetized animals, at least, for sure. But so they just present this thing that they present like gray screen, nothing, as a control. So this is what nothing looks like in the primary visual cortex. Just a gray screen, nothing. And they do this kind of nothing stuff that we call non-natural images. I mean, they call them non-natural images. And then, very interestingly, in another hour of experiment, they go back, repeat the same sequence of images, but without the leak port. So the animal is seeing exactly the same thing as he was here, but it doesn't get rewarded. It doesn't get rewarded because it cannot get rewarded. There's no leak port. It cannot actively respond to the stimuli. I mean, behaviorally, except just watch. Things for which it has been trained to respond and get rewarded, except that it cannot be rewarded. Okay? So, I'll repeat the procedure. We take 30-second windows of activity. We go to momentum space phenomenological normalization group, taking the eigenvalues of the covariance matrix, building this projector, projecting into smaller and smaller dimension subspace. So n over 2, n over 4, n over 8, n over 16 dimensional subspaces. And so this coarse graining, if you do this for all the neurons you have in your 30-second window, will give you something like this. Actually, the result is for these animals from the Allen behavior. Again, so I'm going to compare the kurtosis of the randomized activity with the kurtosis of the surrogate activity. In some cases, you have something clearly non-Gaussian like this. In some other cases, you have some things which are not so clearly non-Gaussian. So is this difference enough? So you have to decide on that now. Okay? I showed you previously this picture from Michael Fisher's review on the randomization group, in which mathematically you do this operation of renormalizing your parameters. Eventually you're reaching a fixed point, which signals scale invariance here. In the model, for the phenomenological normalization group, the way we reinterpret these ideas, or the way we interpret what we are doing, is that you have in this world, this space of distributions, you have some cases like this in which you have some, look, the original distribution is here. Can you see it in light red? The original distribution of activity, and then you renormalize it, you coarse grain, and coarse grain, and coarse grain. Eventually, you coarse grain, and you can go straight to a trivial Gaussian fixed point. But there is another non-Gaussian fixed point, presumably, if there is some criticality in the system, and you can start from, well, a different, in those 30 seconds, your system is here, and you coarse grain and coarse grain. Perhaps you're never going to go there exactly, but you're closer to this point, so that you get this closer to non-Gaussian signature of what's going on. So, in this scenario, the way we interpret where we are in these trajectories by looking at kurtosis. So low kurtosis means that you're tending to this fixed point, perhaps, and higher kurtosis means that you're trying to go to that direction. The question, of course, remaining, in the sense, this kurtosis that you get at a given coarse grain step would amount to some distance to triviality. So there's a lot of talk in this brain criticality literature about trying to find some distance to criticality. I don't have that, but I think I have a distance to triviality. I don't know if the same thing. I think it's not, but it's already something. A distance to triviality. Kurtosis of the coarse-grained activity, and if you accept that idea, that kurtosis of the renormalized activity is a distance to triviality, then the question is, how far must you be from a trivial fixed point to be considered critical? Okay? Like here, right? Is this critical? No. So, what I'm going to do, I'm going to plot a probability distribution of the kurtosis of the coarse-grained activities of all 30-second windows for all the animals. Okay, so I have this. Actually, we do this for each animal, right, Gustavo? So we do this for each animal. So I have lots of 30-second windows. I run the whole thing. I get the renormalized distribution. I take the kurtosis. I keep it. I do this over and over and over. I have a probability distribution of kurtosis of the renormalized, of the coarse-grained activity. Is that clear? You're doing, you get this black curve here. I hope this is clear because this is important. It is? If it's not, please raise your hand. That's okay. It's okay? No? And I do the same for surrogate data. I shuffle the data in every 30-second windows, rerun the whole thing, get something which typically looks closer to a Gaussian, get this kurtosis, and keep it, and then do a histogram. This is what I get. And now I have to decide when, for a given window, which has a given kurtosis K or kappa, whatever, do I label it as this is critical or has non-trivial scaling, or it does not? Okay? So there are different ways of doing this. You can put a threshold somewhere here. Which kurtosis is large enough? Perhaps if you're an over-enthusiastic, gullible person, yeah, here is good enough. That's too, I wouldn't go there. The z-scorer, perhaps. The z-scorer will go here. So there's one mean plus like one or two standard deviations. That would be me. I would be this guy probably. But there are, of course, the guys who don't believe this thing, criticality or scaling or whatever, the skeptic will put their threshold here. And here is the denier, okay? Yeah, nothing will convince him that it is sufficiently non-Gaussian or non-trivial. So how do you do this? Well, what about the recipe for happiness in your life? Try to please everybody. So that's what we're going to do. Try to please everybody. I give you a knob, a control, and you choose whatever the hell you want as a sufficient level of kurtosis. Okay? So I'll come to this histogram here. Let kappa threshold be an arbitrary threshold of kurtosis of the renormalized activity. You choose it. And then I'm going to do all the results as a function of this guy. So you choose a kurtosis which you deem high enough to be considered scaling, and whatever is above this threshold, I'll paint it blue, meaning that it has scaling. Whatever is below this threshold, I'll paint it red as having no scaling. Okay? So for kappa larger than kappa threshold, a window is deemed to have non-trivial scaling. And once you have chosen this guy, you go back to the data, you have lots of 30-second windows, and you painted it blue or red according to your choice. Right? Okay, yeah, this one had scaling, this one didn't, etc. Well, during these 30-second windows, the mouse, sorry, it's not the red, it's the mouse, is watching all those images, leaking, not leaking, leaking, not leaking, Sometimes it gets it right, so these are the hits. Sometimes he gets it wrong, these are the misses. So I have hits and misses along all these windows, right? So the question, before I go to the question, the question is whether, is there any relationship between the window being blue, that is being deemed critical or having non-trivial scaling, and its behavior and its performance? Does it get it right more often when it has scaling, when it does not have scaling? That's a question. In order to control for that, you shuffle hits and misses. You have some, well, depending on how much this is, to which extent this happens, if it happens at all, you have to control by shuffling hits and misses. So I'll show data with trial shuffle behavior in which I keep the classification of a given window, whether it has scaling or not. This has, this one hasn't. But then I shuffled all the trials so that the performance here has no relation to the scaling behavior. This is what I'm going to control against. Now the million-dollar question for us is this: how does distance to triviality, here summarized by the threshold at which you put your choice of criticality, how does distance to triviality impact performance? The plot I'm going to show you is a very simple plot. It's the average hit rate of the blue windows. I take all the blue windows once you've chosen this threshold. I take all the hits and divide it by the total number of trials. That's the hit rate. It's a fraction. Okay? And I do the same for the hit rate for all the red windows. And I compare this with all, so I calculate this for all the windows which are here, above the threshold that I let you choose, as you wish. It's a very simple plot, but one which I like very much. Here's a hit rate. In dashed line, here is the probability of observing a kurtosis in the randomized activity. This gray region is the surrogate data, and the trial shuffle result is the orange curve here, shows it that no matter where you put your threshold, the hit rate is the same, of course, because you're shuffling everything. You're not distinguishing scaling from non-scaling regions, and it's about 70% hit rate, as I told you. But for us, what matters is that if you look at the scaling windows, the ones which have kurtosis larger than the value you chose in the horizontal axis as whatever you want, you put yourself whatever you want the horizontal axis, and then watch what happens to the blue windows, scaling windows, to the red windows, no scaling windows. So higher kurtosis means better performance. The blue curve is above the red curve. And you can calculate, if you want, how much better they are. You take the blue minus the red divided by the red or something like this. So the relative performance gain, just a measure of performance, it can reach, depends on where you put your threshold, up to 24%. Of course, if you put 24% here, you're throwing away most of your data. You're just cherry picking very high kurtosis, but that's your choice. If you do this, you get very high kurtosis. If you look at the typical case, like the peak of the distribution, you get 10% increase in performance. Yes, Silvina?
Sufficiently large kurtosis. If not, you didn't have scaling. So I don't understand the plot, because you have the red curve covering all values of kurtosis. That I didn't understand. That thing that says no scaling, the red curve, I thought it would only hold for small values of kurtosis. That I didn't understand.
The horizontal axis is kurtosis threshold.
Oh, okay.
Right?
Okay.
So I'm letting you choose
Okay.
which kurtosis threshold you prefer. So this is P of kurtosis, which is something like this. This is the surrogate. This is data. So there's some confusion here, which you may or may not want to explore. But say you say, okay, I want to be really larger kurtosis than surrogate data. How much larger? Well, you pick a choice. If you choose like a 9 here, then this is the result. This is the hit rate for all the windows which have kurtosis larger than 9. And this is the hit rate of all the windows which have kurtosis less than 9. Of course, as you increase kurtosis threshold, you're cutting this distribution, having less and less data. But, okay, I mean, it's up to you to decide how much you want a statistical significance or whatever. And so the first critical take-home idea is the following, that non-Gaussian scaling dynamics in the mouse V1 are closely linked with improved performance in a visual task, just therefore supporting the idea that critical dynamics underlie functional advantage in neural processing. To my knowledge, that's the first time that this happens. To my knowledge, I don't know, maybe someone has done this, but not only that, but with a continuous indicator, which you can choose who do you want to include inside or who do you want to exclude with an exclusion criterion, okay? So we were very happy with this result. But there's more. It gets more interesting. This, of course, was obtained, this is performance gain. So we can only measure that while the animal is performing. If it's not performing, you cannot measure performance. Right? So still, this data is very valuable. So the leak port has been removed, but it's nice that the animal is seeing exactly the same images as in the first part, the same sequence. Except it cannot leak. It probably wants to leak, right? Has been trained to leak to those images, but it cannot. So when I go back to the active phase, another way of plotting, making this plot here, probability of observer given kurtosis, is just plotting the cumulative distribution function, or the 1 minus the cumulative distribution function, okay? So the fraction of windows above threshold. So as increase the threshold, it goes down. So this blue curve is representing the integral, the integral from here to here of this distribution of kurtosis. This I can measure here. I can come to this data set and randomize activity, all the brain of the V1 activity, and collect the kurtosis and do an analysis again, and collect the probability distribution of finding a window with a given kurtosis K. Now the animal cannot be rewarded. This is what you get. You probably cannot appreciate the difference if they're too far apart like this, but if you plot them together, this is what you get. So what I argued before, well, the data argued before, was that larger kurtosis meant better performance. Now the animal cannot perform, possibly perform. Therefore, its kurtosis gets smaller. They get closer to surrogate value. Distribution gets closer to surrogate values. Right? The green curve is below the blue one. As though the brain was behaving like this. Well, perhaps having scaling this criticality thing is good for performance, but since I cannot perform, what's the point? Even though it's still watching images for which it has been trained. Therefore, you can go to this highly non-interesting phase of the experiment for the animal, for which it has not even been trained to be rewarded, let alone the lack of significance of the images, which are non-natural. There are no trees, no cats, no nothing, just stripes of things. And then here, it gets even less critical, if you want. It gets even closer to the triviality in the distribution. Right? So this is, I find this incredibly interesting. That's the second take-home idea. The signatures of criticality V1 are modulated by task involvement, by task engagement, but how much the animal is engaged with the task. The mouse brain's ability to self-regulate and display scaling dynamics predominantly while performing a task, again reinforces the idea that scaling plays a role in enhancing sensory processing. So, these probability distributions of the randomized activity are telling us a lot in this experiment. And finally, if you are into exponents, we have exponents, right? So the trivial values are dashed lines here. Alpha 1 minus beta is easier to show 1 minus beta, so we can show the surrogate values here. Interestingly, this alpha and beta exponent, and well, and mu as well, they have been called by this Australian group, right? Static exponents of alpha and beta. They shift closer to trivial values as engagement decreases. So this active phase, passive phase, and non-natural phase, they tend to this more trivial values, whereas Z, which is a dynamic exponent, has the opposite trend. We have no idea what's going on here. If you have an idea, please let us know. We just don't know why they have different trends. So yeah, these 4 PRG exponents reflect differences among the phases and have non-trivial values. Of course, now there are more questions. This was a visual task. So in a visual task, we found clear signatures of criticality in the primary visual cortex. But will we also find such signatures in other regions of the mouse brain? Because the same data from the Allen Brain Observatory, they have that. Other regions, further regions of visual cortex, the visual pathway downstream, and hippocampus, and some parts of the midbrain. So if you go to Gustavo's poster, you can appreciate what's going on. Very unfair summary of a very nice poster with lots of results that as you progress, if you just look at the signal of criticality from these different regions, you can reconstruct the hierarchy, anatomical hierarchy of the visual cortex. And the hippocampus is farther down there. So weaker, the signatures of criticality get weaker and weaker and weaker as you move away from the central stage of the problem, which is V1 in this case. Okay? So this is preliminary results. And another thing that we're working on right now is, are these signatures of criticality innate, hardwired into the system? Or do they emerge as the mouse learns that performing a task leads to a reward? So again, freely available data on the internet from very sophisticated labs, which are releasing optical imaging data, is it? Yes. Calcium imaging, yeah. Of mice while they learn a task. Not only after they have learned like here, but while they are learning. So you can see what learning is doing to those signatures, for instance, right? And another question, we're still looking for people who have the data. Can we see this in humans as well with behavior, for instance, right? This is another thing. In this 5 minutes or 10 I have left, since I mentioned humans, just to briefly comment on a more recent result that has nothing to do with what I told you so far about rats, but it's just that just came out last year. We look at fMRI, RS meaning resting state fMRI data from 714 healthy subjects. So the Human Connectome project makes this available. We run phenomenological renal imaging group on this data and look at the results. And yes, on the whole brain kind of scale, you do find this non-trivial scaling, alpha, beta, and this one is different. Instead of looking at the whole spectrum of eigenvalues, if you just look at the scale of how the first eigenvalue, which is an important one, scales with system size as you renormalize. You have, of course, non-Gaussian distributions. It looks exponential here, like the one Bill showed, I don't know when, a couple of days ago, for the Gaussian fluorescence it was, right? Again, it looks very similar, even though look, one case we're talking about fraction of like a millimeter squared region in the hippocampus, perhaps more, and here we're talking about of a mouse, and here we're talking about the whole human brain. So the thing still gives you signature of criticality, but more interesting, we found this by chance, a little bit by chance, was that when you look at these three exponents, and we just happen to scatter plot them together, they fall into, we notice they're strongly interdependent. They fall into a line in 3 dimensions. And you can fit how they depend on one another. Beta is minus 0.5 epsilon plus 1 point something, epsilon 1 point something alpha minus 0.98, minus half plus 1.56. If you look at those numbers, and you're theoretical physicists, you start to itch because, come on, those numbers look a lot like half integers, and, right? So it could be something there. So we know, I showed before, scaling relations between those classical exponents, tau, tau t, 1 over sigma nu z, they're connected. So this looked like there was some kind of scaling relation among them. And before I go further into that, I just comment that this alpha, and therefore all the other ones, because they are so interrelated, have some positive correlation with total gray matter volume of the patients. So some kind of anatomical score here indicating some, it's just a correlation, but it is a correlation. And there's also correlation with correct responses in some kind of cognitive test. Okay, so it's a behavior score with which data correlated. Physicists try to wave away from correlations like 0.16 and 0.28. Yeah, come on, just a little. Yeah, correlations are small, but significance is high. It's highly significant. And for biologists, they like, this is meaningful, okay? This even small correlations. We make an attempt with some kind of rough model of this data, fMRI data, and somehow can deduce analytically under some approximations these relations between the exponents, which match very well what we observe in the data. And I don't know exactly how to classify the status of these calculations or these results, but they're satisfied by this experimental data, and it's somehow they, then you can revisit all the other results with exponents and see whether they're checked. So in the mouse cortex, this is not exactly satisfied, but it's close to it. Okay, just close to it. So I don't know how this thing will evolve, but I just wanted to mention that because it's a recent result, and after the second week, you're all familiar with this technique, so it could be interesting. I want to thank all these people, very hardworking people, brilliant students and postdocs, and I think we are a happy bunch. You're welcome to join our group if you want, and please let me know. Thank you all for this nice 5 days. Thank you.
Thank you, Mauro. And now it's time for questions.
Hello, Professor. Thank you for the lecture. Very nice lecture. I have a question about a result. So you did the whole PRG for the data on the urethane anesthetized rats, and you saw scaling from a CV bigger than a certain threshold,
Yes.
0.7.
Yes.
Yeah.
Yes.
And you did mention it, but I maybe didn't understand it. So when the rat is awake, it is actually with a CV lower than 0.7, isn't
Thank
it?
you. Excellent question. I forgot to mention that. Yes.
Yeah. So actually, most of the time, the brain is non-critical if you take those results.
Excellent question. We were puzzled by that. So let me try to jump back into the perhaps the most interesting picture. I don't know. Is this one? Forget about everything, just look at this plot, right? This is the plot you're talking about, I think, right? So what this is showing that essentially for CVs larger than 0.7 or so, more or less, do we see a meaningful signature of criticality of scaling. For the urethane anesthetized animal, the thing is, here, low CV means very asynchronous activity, which looks like awake behavior, electrophysiologically. But I've just shown you, I'm sorry, I didn't even mention CV for the awake animals, because they are all small, they are all very, very low. If you go to another poster, Vinicius' poster there, he has like distribution of CV for this whole thing. They're all smaller, much smaller values of CV. It never gets here, or rarely gets somewhere here. So they're all the time around here. And still, with the same technique, it gives you this large kurtosis. So maybe you could forget about CV and just ignore it? I don't know. It's a nice measurement. It's a crude measurement. It's been used all the time. It's useful to some point. To some point. So I don't know how to make this. I don't know what this is a decent project to talk to some electrophysiologists, but apart from PRG, I would be curious to see whether other techniques can tell apart what an asynchronous behavior for anesthetized, urethane anesthetized animal is, and asynchronous behavior from an awake animal. But PRG apparently can. It gives you completely different answers. Look, this may be surprising perhaps to a neuroscientist, but not to a child. I mean, one animal is anesthetized, the other is awake. It should be surprising that they're different, right? That's what I mean. I mean, of course you would expect them to be different. It just happens that this setup of urethane was got so much attention in neuroscience literature precisely because it was considered like an animal model of non-synchronized and synchronized behavior, asynchronous, irregular, and regular behavior. So they thought it was a good model, and perhaps it is to many problems, but not for this one. Thank you for the question. I forgot to mention that. So not all asynchronous states are the same, apparently.
Okay, thank you, Professor.
I don't know how surprising that is. I mean, we were surprised because we were used to CV as it's used for stuff. Well, yeah, no synchronous, asynchronous, low CV, high CV, and we expect it wouldn't work, but it did. So,
Thank you, Copelli, for the lecture. My question is about the another momentos of the distribution CV, for example, scale, or, and they can it can provide another information about the
or there was
critical.
moments of distribution. Oh, sorry, yeah, I forgot to mention that.
For
Forgot, yeah. So in the beginning, I think, I forgot the beginning of the history in our lab, but for some reason we chose kurtosis instead of skewness to look at that. I don't remember why again. Anyway, but so kurtosis worked and we kept with it. But in that model paper that I briefly mentioned by Caio Nascimento, some referee asked the same question. So there are like a possibly infinite number of characterizing how non-Gaussian the distribution is. And we chose, I mean, a bunch of them, and essentially they all work. It doesn't really matter. It doesn't have to be the kurtosis. It can be essentially anything will work. It's just that it's nice to have any number, I mean, the kurtosis or the skewness or whatever, so you can look at the number instead of the whole distribution. That's all. Yeah.
Thank you.
Do we have any other question?
Can we think about learning as a process of improving compression and then make the system activity more resilient to coarse graining?
More resilient to coarse grain. What does that mean?
Like in the plot where you have the flows in the space.
The schematic plot, you mean? Like the cartoon plot? This one? Yeah?
Yes, I mean,
I call this the Cambrainha plot because Gustavo came up with this idea. That was very, very nice. Yeah.
can we think that learning might be a process that moves the states toward the upper curve?
That's our question, yes. That's what we hope to find out by applying the techniques to these other data sets where there's learning.
And do you think that it's related to a process of improving information compression?
I don't know. That's too much. I don't know.
Okay.
I just don't know.
Thank you.
Hey, thank you for the lectures.
Thank you.
In neuroscience, in experimentalist neuroscience, I think there's a eternal problem between temporal resolution and spatial resolution from different means of measure, such as fMRI or EEG. Do you think that with equipment like functional ultrasonography, there has been being talked a lot recently, there could be improvements to criticality hypothesis? Is temporal resolution something that is valuable in criticality or something like that?
Well, the fastest, the more data you have, right? So in a way, yes, but of course you don't want spikes in microsecond times. It will be useless, perhaps, I don't know. I don't know. Another question to which I don't know the answer. But I don't know much about this technique either, so perhaps you can tell me, what is this technique all about?
I've seen work of Daniel Takahashi from Natal.
Mm-hmm.
He's been using some kind of, I don't know how to explain exactly, but ultrasound shouldn't be able to get the waves from the cranium. I was talking to Pedro about it. But he's using it to observe animals like birds, and I think there's some thinner cranium properties that make it possible to get information from ultrasound. But and
You mean neuronal information, neural activity information?
yeah,
Okay.
it's kind of refined. So I was thinking if there's some work in this area, but that's okay.
Fortunately, I'm good friends with Daniel Takahashi. I'm going to ask him. I don't know. Yeah.
Yeah.
Okay.
Thank you.
Thank you.
In the experiment where you expose the mice to the same sequence of images, but in one case you give the reward and in the other you don't, that you observe that when you don't give the reward, the distribution, I think it's the fraction of windows with, that gets like the performance is not so good.
The signature of criticality is not
yeah,
so good.
the signature of criticality.
Yes. So these plots here. Yeah?
That one. Can you analyze the first windows separately in the third case to see if there is a drift? Because maybe if you have enough mice, you could do some statistics over mice, and so just see how this distribution changes depending on the order of the window or something.
Yes. So
whether if there is a drift, that when the mice realize that they don't have the reward, they don't care or something like that.
It's your question looks like the question of one of the, I don't know if it was a good or the nasty referee, but no, but it was a good question. He or she asked whether what we are seeing here, so that the change from the blue one to the green one, were not due to fatigue, perhaps. It's like 1 hour here, 1 hour here, plus half an hour here. So it's a lot of time. I don't know about watching these movies. People do this every day with their cell phones, okay? Hours and hours scrolling. But to these animals with their heads fixed, I don't know. So it's an interesting question. And in fact, it's some kind of supplementary material kind of thing that it is there. There is even during the active phase, please help me here, Gustavo. So even here, if you look at this average kurtosis, if you want, there is some kind of decrease, right? And so there's a point to that. There seems to be something akin to fatigue. But that said, if you stick to this part of the experiment here, for which you have reward, what does that mean? It means that if you look at the probability distribution of kurtosis observed, like in the first phase, say in the first half of the active phase, and the second half, the distribution in the first half, they're more to the right and then goes a little bit to the left. Yes, but when you look at, I'm talking about the dashed curve. But when you look at the hit rate as a function of the kurtosis, as you see, when I put the threshold here, just looking at whatever is above this threshold or not, these curves remain more or less the same. So we can have more or less scaling windows according to your criterion of kappa threshold. But once you choose a threshold and you look above it, it doesn't matter. I mean, the fatigued rat, we have less of those windows, but when it visits those windows, it has higher hit rate. Is that an acceptable answer?
Actually, I was thinking of learning that there was no reward rather than fatigue, but in the third part, but it's okay.
That's our interpretation. That's because there's no reward, right? But then the question, well, what if the rat is just the mouse is just tired? Could be. Yeah. You can imagine all the variations in the experiment that people around the world are coming up in their minds to suggest to the Allen Institute. We has already provided a very nice, incredible experiment. I think Bill had a question, right? No, it's okay. Someone else?
All right, I think then we can, any remaining question, have it in the coffee break, and of course give a bigger thanks to Mauro for his
Thank you.
whole
Guys.
course.
Thank you very much.